to the stage, and the panel then is open for discussion to all the speakers. Okay. So, are there any questions at the moment from the audience? So I would like to start, Professor Tomalia, concerning dendromeres. I mean, you are a pioneer and inventor of the dendromeres. How can you, let me say, guarantee the reproducible production of dendromeres? Is there any possibility to see how the dendromeres are from batch to batch? Because concerning my field, for example, iron oxide nanoparticles, we do see sometimes difference from the batches. So therefore, I would like to ask, how do you see this task?
That's a very good question. And in fact, there have been episodes in the past where dendromeric material sold through Sigma Aldrich and other chemical supply houses have had some problems. We have nearly 25 years of experience, and we have, as you may have seen on that last slide, we do electropherograms of all of our materials, treat them like proteins, you know. And we have some very proprietary but very specialized protocols that allow us to see whether there is heterogeneity batch to batch. And the person running our production has been in the business for nearly 20 years now, and so that's a very good question. We think we have the most monodispersed dendromers in the world right now. And that's what I tried to put on the last slide. And it's by the CNDP engineering. Thank you.
Thank you. Scott, please.
So one thing we hear at NCL is the difficulty in scaling up nanomaterials. So my question for those of you that have some of the more complex, or at least above a small molecule formulation, can you share some of your experiences with just finding a CRO that could produce sufficient material, or are you still doing that in-house for phase 1 and phase 2? If you just comment on that.
We do all of our work in-house, so it's not contracted out to CRO. And fortunately, we've got the experience with the Braxane to scale up to commercial level. And the new agent that I described today, which is the NAB rapamycin, that essentially just fits into the mold, and we use the same process. So that works pretty well for us.
So we have a polymeric system, but we have, I think, in last half a year to a year, successfully upscaled from a few milliliters up to multi-liter scale. And that was done indeed not by ourselves, of course, with the input, how we did it formally in-house, but we outsourced it to a CMO who has really a lot of expertise in the synthetic field. So actually that was the former production side of Organon, Merck Schering-Plough these days. So they have really extensive expertise in the synthetic procedures, which is applicable to our system. So we have tackled that issue and have now really high batch-to-batch reproducibility.
Yeah, so in terms of, and I know Scott, the NCL has also worked with our compound. Our contract manufacturer is actually Polymune. They've got a booth outside, and Pronai's always done everything in terms of a virtual model. So we use contractors heavily to do our work. And I think what's nice about the system that we use at Polymune is that the mixing of the particles happens in a continuous process, and it's allows for very homogeneous particles to be formed, and therefore it can be easily scaled up for what we need for clinical.
Okay. Are there other questions from the audience?
Maybe related to the last speaker, but do you have free DNA left in the preparation, or is it all of it encapsulated?
It's actually at least in our hands, and other people who have tested it, it appears to be completely encapsulated, and it has to do with the amphoteric nature of the particles. It's positively charged during the formulation to help encapsulate the negatively charged oligonucleotides, but then the pH is shifted to physiological pHs, allowing for a net negative. It's about minus 40 millivolts by zeta potential. That allows the negatively charged DNA to be repelled and then can be dialyzed away or diafiltered away during the manufacturing process. And in fact, during our PK, we detect the PNT100, but we need to take the plasma, detergent solubilize it, boil it even before we can release the PNT100. So it really remains as a complex until it enters the cells. And the specificity really comes from the fact that not all DNA is open, so you're not non-specifically killing cells.
I have a question for the last speaker. I think your dose you mentioned was 130, was it, or per meter square? Now that seems to be like a lot of material. Is that include the formulation, or what's the dose of the active DNA?
It's actually the dose of the active DNA is 120 mg per meter square. So it's about 200 mg of DNA that's injected, and there's almost 4 to 5 grams of lipid that gets injected there. So you can imagine that you're really swamping the system, allowing the particle to access many different tissues.
And just to follow up on the safety profile, I know you showed the data, but in terms of specific cytokines and things, do you have data to suggest that? Because clearly with the siRNAs, I think that's an issue. Is it not an issue with the DNA?
We have not seen that. So we, in the phase 1 study, and I'm sorry because I guess I was trying to focus on the phase 2 data, we actually did take serial time points to look at immune markers. So we did protein plexes, and we did not see any increases in TLR markers at all. No increases in IL-6, IL-8, TNF-alpha, none of those. There were increases in immune markers that suggested there was an immune recognition of the nanoparticle itself, but nothing related to an immune response, and we didn't see a link between the TLR markers, which didn't increase, and the fever that we saw. We think that's more of a response as the tumors were dying, cytokines were being released. So it's actually very, I think it's striking in comparison to siRNAs, and it might be because of the fact that they tend to be modified, and perhaps the repeated dosing could cause an immune response. But if you imagine, this is I like to call it a native piece of DNA. It literally, this sequence of DNA is found in every cell of your body, so perhaps it's recognized as non-self, or it's recognized as self, sorry.
So I was, sorry, Mr. Barnes.
I have 2 questions. First to you is the particles you are using, I mean quite a lot of material, they are charged particles. And you are now altogether in phase 1 and 2, probably more than 25 patients were treated?
Yes, I think we're now 30.
Did you see any adverse effect related to what, CARPA, complement activation, you name it? That's question number 1. Then I asked the second lady, but first.
Yeah, so what we're finding is that, any nanoparticle, and especially a negative charge one, will activate complement. So what we do is we consume complement. We do get increases in the CH50, for example, but we don't go in the next sequelae. So in patients, in animals, we saw an increase in the CH3A. There was no increases in this.
But that's a very insensitive measure.
So, but in humans, we consume complement, but we don't get any increases in the later cascade complement factors.
Mm-hmm. Okay, that's good. And I think you have a fantastic system, really. The micelles are really, seems very powerful. But the question is, okay, you have a chemical linkage that binds the drug to the micelle in a way, to this polymeric micelle, and then the polymer and the bond, this bond are degraded, hydrolyzed, By what? Exposure to water? What's the mechanism?
It's indeed chemical hydrolysis, so
Yeah.
based upon exposure, yes.
Okay. So the drug will be released everywhere you have water, basically.
Well, maybe we should discriminate between the drug linker as well as the bonds that are between the polymers.
Okay.
So the drug linker can also be more acid sensitive, so only after internalization you could have the release, while the bonds between the polymer chains, those are indeed chemical hydrolysis based on the physiological pH.
Yeah, but when we talk about tumor, for example, for a long time people believe the pH is very acidic. Now everyone knows it's 6.8, 7, something in this range, maybe 6.6. So it's not that much acidic like we believed in a long time ago. All the recent data suggest about 6.8, something like that, most tumor at least. So you have a delicate way to get a kind of differences between hydrolysis in the plasma and hydrolysis in the tumor.
Yes, I think and we actually see, and maybe you saw also from the PK data, we do have release already after injection into the bloodstream. However, it is to a much more limited extent to, for example, the more traditional liposomes or micelles that really have a higher burst release. So in our case...
Why do you say? I mean, what's the basis that liposome have a burst release?
Well,
Doxil
maybe
has no burst release at all, for example.
Okay.
Ambisome doesn't have any burst release at all. These are the two leading. So
but for at least for
try
more
to stick to the truth in this.
traditional micellar system, those have a PK profile that has not been
That's
optimized versus more the traditional drug molecules. While in our case we have at least more than 95% being conjugated to the nanoparticles for at least the first hour. So then you really allow the nanoparticles to accumulate in the tumor tissue, and there you have the continuous release in time. So I think it's about shifting the release profile. It's not an on-off button that only in the tumor drug is going to be released.
So you mean most of the drug going very fast to the tumor?
You have fairly fast accumulation. Yes, we have also some earlier time points.
Okay, that's very good. I think it's a really powerful system, but
Yeah, you're right.
we'll follow it with great interest.
Chairman, can I ask?
Of course.
Yeah. In this public session, I'd like to ask Professor Tomalia. I have been obsessed by your demonstration of the first to fifth generation of the dendrimer labeled with gadolinium.
Yes.
And she established the size effect, the kidney, the renal filtration, and then liver.
Yes.
So did you ever observe any dose effect in your experiment? If you just to inject 10 times of amount of the dendrimer to the animal, then the hepatobiliary excretion was the same?
I can't say that I have those data in my head at this point in time, but I did that work with Paul Lauterbur back in the early days, and Martin Breckbiel at the NIH, and these data were obtained from many animal studies all the way through dogs. And I don't remember the dosage effects, but these were very reproducible patterns that have been observed in many, many laboratories. And they constitute almost like a nanoperiodic property pattern for these particular entities. And that's what continues to be exciting for me, that you can think about them in a predictable fashion. Thank you.
Yeah, because I'm interested in using lutetium-177 at last, labeled with upon the nanoparticle to be given to the at first the animals and then patients. Yeah. So the difference between the injected dose of those nanoparticles in case of the drug loaded and then radioisotope loaded is just 1,000-fold. So my team is searching for the concentration quantity issues in the literature very seriously and sincerely these days, but I could not find many because everyone was interested in the pharmacological amount of nanoparticles or nanomaterials. So we have a kind of a knowledge gap in that point.
I think it's a good question.
Okay, so I think that Panagiotis Trachopoulos will be very happy that you, Dr. Desai, will shift your system now into cardiovascular diseases, because he was always claiming that cardiovascular diseases are much more common than cancer. You're right, despite the fact. So what do you think? I mean, we do have now a shift in the follow-up of the success in patient. That means after the application in cardiovascular diseases. How do you think how you will manage this to follow up? Is there any flow conditions you will follow up after application of your system in patients suffering from cardiovascular diseases, from plaques or whatever? What do you think? How will be the issues you will address there?
Well, the strategy in the treatment of the plaque or the restenosis is really is a very local monitoring, just like you would do if you had stents or some other intervention like a drug-coated balloon. The amount of the dose that we inject is literally microdose, 500 microgram, for example. At least that's the plan. That's what we did in the pigs and saw a very nice effect. So I think we follow the same procedures in terms of monitoring these patients as you would with any other interventional procedure. In the pulmonary hypertension, that plan still has to evolve. But we know that mTOR is a very important target, and this is a more recent data in the last year. And it's shown that one of the primary causes for the progression of pulmonary hypertension. So these patients get worse and worse and worse over time, and the only treatment that they have is really symptomatic treatment. So there is no drug that is used today that can reverse the disease. It is a proliferative disease. So it's often called cancer of the blood vessels, as you know. So we hope to be able to address this using a very specific target that's present in the biology of this disease.
Okay, so if there are no further questions, I would like to thank the speakers once again for their really interesting and exciting talks. And now I think it's lunchtime. Enjoy the food. Thanks.